Hi, everyone. Happy Wednesday. Tonight, we are doing a very special ILADS panel with a number of different experts and providers that serve not only the tick-borne disease space, but the IACI space. So, no matter what condition you are tuning in to learn about tonight, you will probably walk away with some major knowledge. So, a little bit about ILADS first, just in case this is your first time ever hearing about ILADS. ILADS is a nonprofit international multidisciplinary medical society that is dedicated to appropriate diagnosis, appropriate treatment of Lyme disease and other associated illnesses. ILADS promotes the understanding of Lyme and associated diseases through research, education, and policy. They are a major advocate and educator of physicians, of scientists, of researchers, and all different types of healthcare professionals. And I can say, as an 11-year Lyme patient myself, that has worked with a handful of ILADS trained doctors that have gotten me back to health, that they are dedicated to advancing the standard of care for people living with Lyme and other IACI conditions. So we are so lucky tonight because we have the hosts of Tick Bootcamp, Rich and possibly Matt, joining us to moderate this panel. If you have not followed Tick Bootcamp on Apple Podcasts or Spotify or Instagram, I might be biased, but I think it is one of the very best podcasts in the space. So with that, I am going to turn it over to Rich and let our panelists introduce themselves, and he'll take it from there. Thanks, guys.
Thanks, Ali. It's an absolute pleasure to be working with ILADS again, and we at Tick Bootcamp consider ILADS to be one of the best not-for-profit organizations in this space. And we think they're probably having the greatest impact in large part because they're training the doctors who are treating the patients in this community. And as much as I appreciate all the not-for-profits and all the great work they're doing, I don't think we can lose sight of the fact that ILADS is really doing the best work in training the people who are treating the people who are suffering from these diseases. And today we actually have four of the stars in the Lyme disease community. And I can say that without hesitation because I've interviewed each of these people several times at different times, both on long form and short form portions of our platform. And what we're going to do tonight is we're going to start with Dr. Chris Winfrey. I'm going to talk a little bit about him before he begins his talk. And after Dr. Winfrey, we're going to interview Nicole Bell, who we call the Lyme disease engineer, and we'll talk a little bit more about Nicole's background and how she's become one of the leaders in the space. Then we're going to interview Dr. Tanya Dempsey, the person who melted my brain at this year's ILADS conference, and we'll talk a little bit more about that and what cool, how we're going to frame Dr. Dempsey's contribution. And then we're going to talk with my favorite young doctor from last year's ILADS conference, Dr. Melanie Stein, who is going to talk with us about cellular wellness. So, last year, I actually met Dr. Chris Winfrey at the ILADS conference. He was actually my favorite interview last year. I actually did not know Dr. Winfrey before last year, but he was my very favorite interview last year. And as it turns out, he wasn't just my favorite interview last year. He was our community's favorite interview because his short form interview was actually the interview that received the largest number of downloads from the Tick Bootcamp podcast. So Dr. Winfrey is a psychiatrist by education and training. He is the medical director at New Image Wellness. He is always very well prepared. What I like about Dr. Winfrey is he's approachable, but he's also very serious and always very well prepared. And I think our community really appreciated that last year. And actually, he and I had a great interview, which will be released on the Tick Bootcamp podcast from this year's ILADS conference. And we're going to start with Dr. Winfrey because, in my view, the brain is the most important part of I guess, us. It is what controls everything from the minute we are born to the minute we die. And I don't think there's enough focus on the brain generally in the physiological and the emotional elements of the brain in the Lyme disease journey. So that's why we're starting with Dr. Winfrey. So, Dr. Winfrey, why don't you first introduce yourself to folks if I didn't do a good enough job of introducing you? And why don't we introduce the brain generally and what the physiological presentation of the brain is?
Well, thank you for a wonderful introduction, and I'm glad to be here with the rest of the panelists and with the larger community that's listening and watching as well. I don't have any more to add to the introduction. I think you did a wonderful job.
Well,
Thank
thank you.
you. I appreciate your thoughtfulness and kindness to me always. Um, so you prepare some questions, and I'm gonna stick to your questions as much as possible. Uh,
Okay.
and to some degree, w why we should be thinking about the brain and the nervous systems is particularly because Lyme is very fond of the nervous system, and it becomes in some ways the central battleground that we have to contend with when we're thinking about this illness and when we're thinking about recovery. The brain is uniquely vulnerable because it's highly immune. It's a high energy demand organ, and its delicate electrical and chemical signaling is very vulnerable to Lyme and this trophic nature and this toxicity that it produces. Um, and to some degree, I think it's best that we not just think of Lyme as an infection, but as a neuroimmune illness. And I think when we put it in that sort of context, we can better appreciate its impact on the nervous system. Um, and I think that to some degree, the nervous system also has a special sort of place because it's difficult to measure in a lot of ways. It's not very vulnerable to a simple blood test. And I think that is kind of what confuses a lot of people when you're trying to get some sort of accuracy. How do you measure or approximate to what's really going on? How do you get the best measurement? And what are those sort of instruments to use? And I think there's some of the challenges there and why it's a little bit more mysterious than perhaps other ways of thinking about the body. Um, I think the other sort of thing when we're thinking about other organs, we think of other organs in terms of symptoms. And the way we think about symptoms in the brain is much more about network dysfunction. And so the brain is an organ of a lot of different networks that begins to go awry when Lyme enters into the system. And so to some degree, Lyme is very sophisticated in knocking out most of these sort of networks. And so some of these networks that people may not have heard of these sort of words and terms because most of the times when we're thinking about the brain, we're talking about neurotransmitters and things of that nature. But those neurotransmitters are in a sort of array or a constellation that they're regulating networks. So a couple of those networks are the default mode network, um, the Cellius network, central executive network, the limbic network, the autonomic network, just to name a few. And I'll throw another one out there, the action network. So the default mode network is a network that allows you to do a lot of internal thinking and reflection. The Cellius network is the network that allows you to toggle between the different networks. The central executive network is the network that allows you to organize and plan things. The action mode network is to allow you to execute what you plan. The autonomic network is the regulatory network, and then the limbic network is the network that's regulating threats and fears and things of that nature. And so Lyme has a way of impacting all of those different networks, and the combination or the constellation of them, if we can kind of identify, is variance from something like Bartonella or Babesia, for example. Um, and so because of the diffuse sort of impact on all these networks, it can what leads to many people say, I feel like my brain is failing, or I feel like I have dementia. It just seems like there's a total brain shutdown when this thing is really raging at its height. Um, so I think a couple things for us to be kind of be thinking about is how do we move for many people to thinking about, for example, mental health and moving more toward brain health and whole body health and not just kind of the abstract ideal of what the mind is. And then how do we move from its age-old sort of influence, which is the catecholamine hypothesis, where we think about neurotransmitters, how we move from that to network-based thinking and how we understanding the brain and the nervous system. I think we kind of approximated a better understanding. We kind of move into that paradigm, and then we can better understand what Lyme is doing and how it's impacting the brain and why it's so hard to think and to function, to organize, to plan, et cetera, when Lyme is at its height.
Okay, so I'm going to ask you to pause there for a second, and let's talk about let's define humans as spiritual beings, as emotional beings, and as physical beings. And if we're going to define a human in those three general categories, can you talk to us about how the polymicrobial infection that someone would suffer is now going to have, and we'll bookmark the spiritual piece for a moment, and let's focus on the impact that this polymicrobial infection is going to have on us physically and emotionally, and why is this polymicrobial infection going to do that? And does it necessarily have to cross the blood-brain barrier to do that, or can there be other reasons why that is taking place?
So if we're keeping it kind of restrictively to Lyme, it certainly does cross the blood-brain barrier. It's it crosses into every organ in the body. And that way is very unique from some of the other infections that are kind of, to some degree, trapped to blood cells, like Bartonella is restricted more to red blood cells and endothelial cells, or sometimes some of the macrophages and things of that nature. But Lyme can get into any organ and into any cell, and this ability to cross over makes it a lot more dangerous in that respect. But it doesn't only just move through cell membranes and damage cell membranes and damage neural tissue, but also does it to the blood flow system as well. It also does it by destroying myelin and things of that nature. So what we can see at a very dysfunctional level, we can get a blood flow issue, we can get a nervous system synaptic connection issue, and that combination is really problematic. When we think about it at a physical level, when you damage any of the physical tissue of the nervous system, you're going to automatically begin to generate emotional issues. And I think one of the maybe difficult things to kind of grasp is that emotions are somehow non-physical. They are very physical. Feelings are the abstract thing. So feelings are the interpretations of your emotions. Your emotions are very physical. We can measure emotions, and we can put a person under a PET scan and we kind of measure how you feel. We can show you pictures and things and generate certain feelings in you, and we can map out what you're emotionally experiencing. But the way you interpret that are what we call feelings. And so that automatically becomes impacted when Lyme begins to destroy the functions of the nervous system. And so we can see that play out. So we have both a physical and emotional problem going on at the same time. They're one and the same. They're not separate.
Okay, so let's talk about the physical piece first, and that is, are we going to define the brain as that the organ that allows us to interface with the world? And if we were going to do that, is the brain going to be the organ that is going to allow us to sense objects in the world, and after sensing objects in the world, perceive those objects, and after we perceive those objects, control how we think, how we feel, and how we act from a physiological standpoint, and connect that to the impact that the brain will have on the immune function when we are dealing with the inflammation and or the polymicrobial infections crossing over the blood-brain barrier?
So I would say the brain is not the organ that's interfacing with the world. I would say that the brain is the organ that's organizing impulses and stimuli that's coming in from outside the world and
Okay.
inside the body, and that interface or exchange. So the brain is organizing all the information. So it's in the territory of the central nervous system. So it's centrally regulating and organizing all the information. But every part of our body is interfacing with the world, from our skin to our immune system, is collecting all kind of precious data and information and trying to process it and put into some meaningful information, both consciously and unconsciously, for us to move about in the world. Why that is important is because Lyme disrupts all of that. its ability to disrupt the immune system, hijack it or suppress it in different ways, impacts our ability to unconsciously read things. So when we're in facing dangers, so many dangers our body and faces all the time, we're not conscious of all of that. So our immune system is picking it up unconsciously, processing it, dealing with it, whether we're even aware of that or not. And so um, Lyme has a way of interfering with that. Our sense to how Lyme interferes with the gut function. So when we talk about having a gut feeling, for example, um, it's this sort of sixth sense, this sense of getting information about something that bypasses consciousness, this sort of intuitive realm. And if you, in some ways, have a very dysfunctional gut, what comes as a byproduct of that is having intuitive dysfunction. It's hard to read, it's hard to sense things. And so people tend to pick up information and doubt it or struggle to interpret it. And so people get into a situation where they might say, people who have Lyme might say something like this. I had a feeling about that, but I just kind of downplayed. I kind of doubt. I didn't think it was the right feeling or whatnot, or maybe it was so suppressed you didn't pick it up at all. And so I think that these are ways in which Lyme interferes with these sort of nervous impulses and how we pick up information and interface it with the world, and why sometimes living with Lyme is so difficult to navigate the world because you don't have all of your sort of interpreting or sensory apparatus that allows you to pick up information and to make decisions.
Okay, I love the way you describe it. So we have all of these sensory tools. Our we have pressure sensors for feeling. We have taste sensors. We have visual sensors. We have
Mhm.
hearing sensors. We have all of our five senses. And what's happening is our brain is organizing all that data and creating a perception in our brain. And now, because our brain is not functioning as a result of this disease, we are now not able to organize information, and we're not able to create perceptions in our brain that are necessarily consistent with what will serve us in our interfacing with the world. So now let's talk about the emotional piece and the cognitive psychological profile of beliefs triggering our thoughts, thoughts triggering our feelings, feelings triggering our actions, actions giving us our results, which then reinforce our beliefs. And let's talk about the impact that belief has on our capacity to first define our illness and then define our capacity to heal.
Yeah. One of the things I think is so important is this relationship between infections, toxins, and emotions. And so they all three are in relationship together. And when we're treating somebody, we need to be thinking about all three together. Um, they kind of hang out together at a very energetic, similar frequency. And so when you're trying to treat the infection, you're not treating the toxin as well, you can kind of get into a disequilibrium and can make it very difficult for the person. But it's the same thing with not dealing with the emotional component. It's sometimes hard to get rid of Lyme because the thought and emotional component is out of balance as well. And so when we really put this into perspective, the studies show that if you are negatively thinking, if you are nihilistic, if you're extremely doubtful, if you're pessimistic, these sometimes thoughts and feelings generate very strong immune dysfunction. It generates a lot of inflammation, and it can make it very difficult for your nervous system, such as your autonomic nervous system, your stress system, to kind of regulate things properly. And so, having the right frame of thinking is really important to being able to successfully heal. So, to some degree, when we're talking about autonomic nervous system balancing, those that kind of, let's say, are type A, and they're in a lot of chronic stress and fight or flight sort of experiencing, and they think that they're just going to fight their way to the healing place. It doesn't work. It's an antithetical sort of thought process to healing because healing is under the umbrella of the parasympathetic system. And the parasympathetic system is asking us to rest, is asking us to experience peace, it's asking us to digest, it's asking us to metabolize, it's asking us to grow. And that's a different thinking process. And so the constant fighting that sometimes people have doesn't lend toward healing. The only way, the only sort of thought process that gets us into the healing pathway is an attitude and thought process of submission and surrender and acceptance. And for people who are oftentimes type A or people who are going through a lot of trauma, they struggle here. And because they can't get into that sort of place and thinking, they can't adequately get into the parasympathetic, then you can't override the sympathetic system, and then it just things running amok, and healing becomes extremely difficult.
Okay, so let's build this out, and I could have this whole webinar with you alone, Dr. Winfrey, but we are going to have to talk with some other folks today. So let's build this out. So we have a binary brain, and our binary brain is one where we are either going to have a sympathetic expression of our nervous system or a parasympathetic expression of our nervous system. We're either going to be fight or flight or rest or digest, as you were defining it. And of course, when we're in the sympathetic expression of our nervous system, or fight or flight, what it does, and I'm going to talk with Dr. Dempsey a little bit more about this as well, but it shuts down our immune function, or certainly limits our immune function, because what our brain is doing is it's trying to pull together all the resources necessary to help us to survive, right?
Yeah.
And when we get stuck in that loop, we are in a place where we can't physically heal, which is why the belief patterns are so important there. So can you build that out for us?
Yeah. I think that when it comes to healing requires a few different beliefs. One, it requires faith, it requires trust, it requires submission, it requires surrender, it requires my mind just went blank on that one, so I'll stop. And so submission in some ways is the ideal that one is coming under what the mission is. And then surrender to some degree is the idea that I'm not going to keep fighting and controlling the situation. I'm going to trust that it's going to work out, and I'm going to walk the path within parameters. And I don't know exactly how I'm going to get there, but I'm trusting I'm going to get there. I'm going to stop fighting the path, and I'm allow things to happen within me instead of me trying to dictate every aspect of it. That is in some ways the sort of thought process that is needed for this direction to go on.
That's a perfect way of winding down this portion of our talk, because now I'm going to pivot over to Nicole Bell, and we're going to talk Nicole. We're going to talk with Nicole about a number of different things. And one of our friends, Nicole, has been interviewed by us here at Tick Boot Camp many times, and it's really been wonderful to watch Nicole develop a very different purpose. And for those of you who don't know Nicole, and please listen to our long form podcast to get more information about Nicole and her journey. But unfortunately, Nicole's purpose changed after her husband became ill and ultimately died from Lyme disease. And that became a defining moment in Nicole's life, which began with Nicole taking her diary and converted it into one of the most powerful Lyme disease memoirs I've ever read. And Nicole is now the CEO of one of the top labs in the country. I really we at Tick Boot Camp have certainly been big fans of Galaxy, especially with the work that they had been doing before Nicole joined them in the Bartonella space. And we also particularly love that lab because it was founded actually by a veterinarian. And we feel really strongly at Tick Boot Camp that we have to make sure that we're focusing on the whole spectrum of Lyme and the whole process. And the founder of Galaxy has given us, again, another brilliant man who's been a wonderful guest on our podcast, gives us a very different perspective on what's happening with the various germs when they're being brought from the animal host over to the human host. So Nicole, please say hi to folks, and let's talk about a couple of things. First, I'd like you to share with folks the wonderful paper you had written on Lyme disease. As you know, it is the paper that we recommend to all of the people who are newly diagnosed with Lyme disease. And let's talk about testing after that.
Sure. So, hello. Thanks for having me here. It's a pleasure. And for all the people listening in, really happy for you to be here with us tonight. So thanks for describing my background. I think that the paper that you're referring to is the paper that I worked in collaboration with the Center for Lyme Action. So prior to joining Galaxy, Bonnie Crater, one of the founders of CLA, read my book, and she asked me if I wanted to take on this project where we summarize the state of Lyme disease research. And it's a daunting project, right? Because there's been so many advancements that have happened in the last, decade or so, really digging into the really just focused on Lyme, the physiology, how it impacts cells, and then how it's linked to complex chronic illnesses and why it occurs and how it evades the immune system. And so they wanted to summarize that in something that they could use with congressional staffers and lobby for more funding for research, right? Because there's so many things that we still need answers to. And so that paper's available on the CLA website. We've done a couple of webinars on that with CLA, and there was also an ILADS webinar that I believe is available on the ILADS site, where we go through some of the papers and talk about the research. And I think we've got an update coming up with another ILADS webinar in the spring, where we talk about the research that's been released since we launched the paper. And so that's a resource that I think is good for physicians, but it's also good for patients and caregivers. It's something that I wish that I had when I was dealing with my husband's disease, because it's so complicated, right? There's so many things that you need to understand, and it's really sciencey stuff, which some people don't have, all the background that they need to fully understand. So I would definitely encourage people to check it out.
So I can't recommend that paper enough, and I would describe it differently. I think it's a paper that is broken down in a way that's understandable to lay people. And I know the purpose of the paper was to define the state of Lyme research, but what I really liked about it is it's a really easy read, and it's really easy to understand, and it defines Lyme you walked a very I think careful line in even defining Lyme disease. So it's really, really well done. And quite frankly, I like that it was written by an MIT educated engineer. I think it was a very, very nice presentation of this disease. So Nicole, the main thing I wanted to talk to you about today is the state of diagnostic tools. And the reason I thought we should transition from Dr. Winfrey and the conversation that we were having about emotions and the importance of belief is because what we've learned at Tick Boot Camp is that Lyme disease generally strips people of their identity. And one of the processes that causes people to lose their identity or lose their sense of self is that they start to believe the lies that they're told as a result of the difficulties that come along with diagnosis. And what happens in many cases is a Lyme patient will go to their doctor, unfortunately, in most cases, not an ILADS trained doctor. And because they're going to their doctor and they're describing these migrating symptoms, the doctor starts to disbelieve that the person is in fact sick. And then what happens in that case is the doctor uses whatever testing is available. And Dr. Dempsey and you are going to help us both talk about why the state of testing is so bad. But in most cases, the tests will come back, which indicate that someone does not have Lyme disease, which then causes their family to believe the doctor and disbelieve the patient. And because the family now doesn't believe them and their support system doesn't believe them, they now start to, again, lose that connection. And then the worst part of it all that we've observed is people begin to doubt themselves. And I think all of this is a function of not having well-trained doctors, and even more importantly, not having diagnostic tools that we can rely on. So I would really appreciate if you can share with the folks on this webinar the different types of direct and indirect testing and some of the great things that you're doing at your lab.
Yeah, so testing is so key because so many medical decisions, reimbursement, all the things is all based upon having a lab result that says, I have XYZ, right? And if you have a test result that says you don't have XYZ, then you're not going to get covered. You're not going to get treated. You're not going to get believed. And that was our experience, right? Our first, treatment, or first visit to the doctor, I actually thought, hey, I want him tested for Lyme disease because it seems to fit all of the symptoms. And we tested him and he tested negative. And then they referred us to a neurologist and he got his Alzheimer's diagnosis, and that's a whole story. And so, the misdiagnosis is rampant. And so why is that, right? Why are the current standard of care so inaccurate? And it's essentially the basis, the standard of care is based upon what's known as indirect testing or antibody testing. And this is a method that relies upon your immune response to behave properly and to have a high level of antibodies that can be detected by the test. The reality of Lyme and also some of these other pathogens, because we talk about Lyme, but Bartonella, Babesia, some of these other infections are equally as sneaky, is that they actively evade the immune system, and they have a whole bag of tricks of ways to suppress the immune system, hide from the immune system. And we talk about this in the paper, and I saw a couple things in the chat adding what's the name of the paper. It was literally the state of Lyme disease research, and you can find it on the CLA website. But we talk about all the ways that Lyme evades the immune system. And then what do we use to determine whether or not you have it? An immune marker. You're an antibody, right? So it's a fundamentally flawed approach because, and even in a lab environment, one of the studies that I love is from Dr. Monica Embers, where she looked at the same bacteria in rhesus monkeys, the same strain of Borrelia in two, rhesus monkeys. And one monkey has a very robust immune response that persists over time. And then the other monkey has a little blip and then nothing. And both were infected when they did the necropsy, right? So even in a controlled environment, the immune system behaves very, very differently. And now we have humans, right, with different infections, different comorbidities, different environmental exposures, and we expect the immune system to behave in this very way. And it just leads to all sorts of misdiagnosis. And the data that's out there shows that over 50% of early patients get misdiagnosed, but there's also a lot of late stage disease because somebody like my husband was sick for a really long time and his immune system was suppressed. So he didn't have antibodies because his immune system was so dysregulated. And then some people actively take immune suppressants because their doctors give it to them and say, oh, you feel bad? Here, take, a steroid to suppress this immune response. And so there's all sorts of things that can bound the results. So that's an indirect methodology, and it's a useful tool, but it's limited, and it can lead to a lot of misdiagnosis. So what drew me as I worked on the CLA paper and then I started researching because as you mentioned, I'm an engineer, right? I like to solve problems. And I'm like, that first lab result that we got that set us on that wrong path, I'm like, what the heck, right? Like, how can we fix that? There's got to be better solutions out there. And direct detection is really used across all infectious disease, right? And what do I mean by direct detection? So just think about COVID, right? If you want to know if you have a COVID test, you don't get, if you have COVID right now, you don't get an antibody test, right? I mean, I have antibodies. I've had COVID. I've been vaccinated for COVID. And so if I want to know if I have COVID, I get a PCR test or an antigen test. But the problem is that these pathogens are stealthy, right? They're low abundance. And so getting them and finding them in a lab sample is really tricky. And so if you use traditional methods, you're going to come up negative even if you're actually positive. And Dr. Breichwert, who you mentioned, one of our founders over at NC State, who really helped put Bartonella on the map, he's a brilliant scientist, but he's also a farmer. So he uses great analogies. And his analogy is, if I'm in a boat and I take a bucket and I put it in the ocean and I look in the bucket and I don't see a fish, does that mean that there's no fish in the ocean? No, it just means you didn't capture one in your bucket, right? And so one of the things that I liked about Galaxy's approach is trying to figure out what are the ways that we can make direct detection actually work. And using sample enrichment and using advanced PCR techniques to really get better detection methodologies. And then also understanding the biology of the pathogen. So for example, Lyme doesn't like to be in the blood, right? It hides in tissues. And so blood is not the best sample to detect Lyme, but urine actually is. And why is that, right? Because the bacteria actually sheds lots of proteins, either as an immune evasion technique or as part of its replication cycle. It sheds its protein code. And then those proteins, which are antigens, essentially gets filtered through the kidneys and into the bladder and excreted in the urine. And so partners at research at George Mason University and Series Nanosciences actually took that and said, hey, we can capture these proteins, concentrate them, and then detect it. And that's a much more reliable methodology for detecting Lyme disease. And so that's the approach we use for Lyme. And then we have a blood-based test for some of the other pathogens, for relapsing fever, Borrelia, Bartonella, Babesia. And as a result, we've been able to really uncover a lot of these cases that were missed by kind of traditional approaches.
All right. So, Nicole, talk to us about what's on the horizon and how you folks are using AI to assist you with some of the tools that you're developing, and how this increase in our capacity to be properly diagnosed will help us to have better relationships with ourselves, better relationships with the people who are in our support system, and better relationships with the doctors who are treating us.
Well, I think one of the keys in this disease state is that you have really complex pathogens with a lot of overlapping symptoms, right? And so it's really difficult to discern. And we all know that a lot of these pathogens, because of the limitations in diagnostics, it's been a clinical diagnosis. But I also understand that making that clinical diagnosis is really hard, right? So our goal is to provide the best possible testing so that doctors don't have to guess, right? And patients don't have to guess. And I cannot tell you the number of times. So we recently launched what we call as the BBB assay. And this is a genus level test, meaning it covers all species for Borrelia, Bartonella, and Babesia. And why is that important? And why do you include Borrelia? Because you just told me that Borrelia is not in the blood, right? So Borrelia is not in the blood, but Lyme Borrelia doesn't like to be in the blood. But relapsing fever Borrelia is actually a different species caused by different species, and that is in the blood, particularly during febrile episodes, and even in non-febrile episodes, you can find it in low abundance. And so we do that, and then we have Borrelia and Bartonella. And I can't tell you, since we've launched this test, how many times we've talked to a provider and they're like, I know that it's Bartonella. Like, this person has all the Bartonella symptoms. And then we run the test, and we also run our Bartonella serology, and it's completely negative for Bartonella. And guess what pops up? Babesia. Right. And it's like these symptoms and some of these pathogens are so sneaky, and there's a lot of symptom overlap, and then they often come together. So we just recently had a case, that we worked with a provider on it, and this person did have Bartonella, and they were treated for Bartonella for years by multiple providers. And essentially, what ended up happening when we tested is the Bartonella, there was no indicators for that. But we did find Babesia, and that had never been identified in previous lab work. And that was what was holding the patient back. And the provider told us, for the first time, every time he tried to treat this patient, they had a negative reaction, right? And as soon as they started treating for Babesia, he started getting better. And it's like, they come together. The Babesia and Bartonella are often like, cahoots, they're in cahoots. And so you really need to be thinking about it. And we've had the reverse, where somebody's convinced that it's Babesia and then it's Bartonella, or they're both there. And so it's really complicated. So our goal is to provide the best possible testing methodologies so that doctors don't have to guess, and they don't have to make the clinical diagnosis, because the symptoms are so difficult to discern that it's easy to be treating for something that is really not the root of the problem.
So now we're going to move on to our immune function expert, Dr. Tanya Dempsey, and I want to share a couple of things with folks. So, Dr. Dempsey, of course, is the medical director. Is it Ames Center, Dr. Dempsey?
Center, yeah, that's right at the Ames Center for Personalized Medicine.
And another one of our panelists who has gone to some of the top colleges and universities. Is it Cornell and Hopkins? Is that
Yeah.
your educational background?
Yeah.
So I do have I'm old, but I have some memory left. And what I want to share with folks is that Dr. Dempsey, I interviewed Dr. Dempsey several times, and she was my favorite interview at this year's ILADS conference, because she answered so many questions that I had about immune dysfunction. Dr. Dempsey has also become a star generally. She's been on some of the top podcasts, not just the Tick Boot Camp podcast, but some of the top national podcasts. She was on Dave Asprey's podcast. I listened to that podcast. It was really well done. I was a little embarrassed about how much better he was at interviewing folks than me.
No.
But you did a fantastic job there, Dr. Dempsey. So can you talk to us first about the immune system, talk about mast cell, and talk about now, fill in now some of the questions that people have about why indirect testing fails. That was the thing that caused you to melt my brain during our short interview at ILADS.
Yeah, and I'm so glad Nicole sort of set the stage. So that's great. Okay, so we're talking about these pathogens, we're talking about these infections in our body, but what is going on in our body that has to deal with these infections, right? And that is our immune system. And part of the reason why people don't get better is because, again, it's affecting a lot of systems. But if we're talking about the immune system is the first line to treating infections or dealing with infections. And you basically have like two parts of your immune system. Um, the the primitive immune system, we call it the innate immune system, and then what we call the adaptive immune system, or like that's the antibody system. And what your immune system does is the minute it sees danger, and that could be from an infection, an acute infection. It certainly also could be from chronic infection. It gets kind of confusing. The innate cells, including this one cell that I talk a lot about, which is the mast cell, is the first line of defense. So it sees the infection, it's trying to deal with it, and it essentially explodes and releases these various chemicals, which are very inflammatory. Mast cells, some people might know, they're related to they are the cause of allergies. So they release histamine that causes lots of inflammatory symptoms in the face, like sneezing and runny nose and itchy eyes. But these mast cells are everywhere, and they're really going to respond to, again, we call it really this environmental soldier. It's really looking for what is bad, and tries to fight it. It then recruits the rest of the immune system to come in and either produce antibodies or do other things to try to get rid of what it sees. What happens with these infections is that, as our other panelists have referred to, these are kind of stealth. They camouflage, they suppress the immune system. In one way, actually, they activate the immune system in other ways. And so it becomes really a mess in trying to deal with it. And that's why, unfortunately, for many patients, it becomes a chronic problem. These mast cells, which everyone has, whether they have Lyme or not, become dysfunctional in people who have chronic infections. They continue to stir the pot. They continue to release at a low level these various chemicals. There are over 1,200 chemicals that mast cells can release, and they're all inflammatory. They're cytokines, they're enzymes, there's so many different categories, and wherever they're releasing, whether it's in the GI tract or the respiratory tract or the skin or the GU tract or the nervous system, they're going to cause inflammation. And so what often happens with chronic infection is that there's this state of alarm from the innate immune system, but the other part of the immune system, which is the more chronic or adaptive, can't function properly. And so that's why often patients have trouble mounting an attack. That's why when you do a test looking for antibodies, you can't find them because they're not being produced properly. Or, okay, here's what blew your mind, I think.
Absolutely, you're getting there.
Okay, or the opposite, it actually sends a abnormal signal to that part of the immune system and says, oh, actually, it's not just Lyme, but it's actually also 10,000 other things and viruses and all these, and thyroid and ANAs, and all of a sudden it's telling that part of the immune system, you've got to, make these crazy antibodies. And so when you test, these are patients who have every band positive on the Lyme Western blot. They have positive thyroid antibodies, but they don't have Hashimoto's, but they look like they have Hashimoto's. They don't have any symptoms of it. They have positive ANAs. They have all these like crazy antibodies. And that's the dysfunctional immune system causing it to go haywire. So it's obviously a really, really complicated situation. And I think what's really important to think about is when we think about these infections, a lot of times within ILADS and elsewhere, we talk about killing the bug. We're always thinking about how are we going to, what's the next new treatment for what antibiotic, what IV, what's going to kill them. And while I'm sure that is still important, and I'm not going to minimize that. I think the root is fixing our immune system, which has really become so dysfunctional because of all the environmental factors that we've been exposed to. the mold, the plastics, the plus pesticides, I mean, the list goes on, right? The forever chemicals, I can keep going. So, like, we are in a state in current times, unfortunately, of already sort of a heightened immune response. Then you get exposed to these vector-borne infections, and then boom, you're in this state of chronic inflammation and chronic infection. And so, for me, my approach is sure, I have to deal with the infections, but I also really, really need to get that immune system to function more appropriately so that the infections can be cleared, or at least
Okay.
the load
So
level.
it's always the immune system that's going to win the day, right? I mean, we just become toxic in some cases because of the germs that have been spit into us from a tick bite. In some cases because of germs we've been picking up during the course of our lives, and in some cases just lifestyle, plastics. I mean, we can't avoid them, right?
And stress,
you know,
and sort
and then of course
of,
we have the Dr. Winfrey piece, which is
yep.
why we need to both make sure that we are focusing on our emotions so that they're not suppressing our immune system and the toxins that we're carrying so that doesn't interfere with the immune system so our immune system can manage the germs that it had been managing before we became toxic and it overwhelmed our system, which is why it's so great that we have both, you and Dr. Winfrey here talking about these two different pieces together. So one of the things that I really enjoyed about our talk, in addition to you melting my brain about how our immune system will in many cases I mean, creating antibodies for an illness we do not have. That was just so powerful. But talk to us about some of the cool tools you're using to modulate the immune system and the success that you've been having with that. That was such a really cool part of our conversation as well.
Oh gosh. Yeah, I do. Well, I do a lot of things. So like, the simplest things are really just um, addressing and stabilizing these mast cells, right? So there are a number of supplements, drugs, and therapies that are focused on helping the mast cells calm down, stop releasing all these different inflammatory chemicals, stop the inflammation, right? I don't want to, I say simple, it's not that simple. There's a whole process for it, but that is something that is I guess, available to a lot of people. You can get antihistamines over the counter. There are a variety of different things that can be done. But what I'm really interested in is because unfortunately that doesn't always help. The medications that we have available can definitely make a big difference, but they don't always. And so if the immune system is still recognizing there are things that are bad there, right, you have to go in with some other tools. So the two tools that I love, and we're just seeing really great results, and I hope to publish on this because it needs to be published, is the use of plasmapheresis, therapeutic plasma exchange, and another therapy called supportive oligonucleotide technique, SOT, or I guess now it's called Q restrain. And so I'll start with the SOT first, because it's really a molecular treatment for these infections. It's, let's just say you have Bartonella. The lab can identify the DNA of your strain of Bartonella, create a small little protein or an RNA that matches the DNA of that Bartonella, and you get it infused. It's a very actually simple thing. It's an infusion, and over the next 4 to 6 months, that oligonucleotide, that little protein, is binding to various Bartonella cells and telling it to stop multiplying. To me, the fact that we can target it in that way is really incredible. Whereas when you use antibiotics or you use herbs, it's very much like a, sort of a wide net approach. Like, hey, we're just gonna throw everything at it and hope it sticks. Whereas this is targeted and targeting the various infections that people have.
All right, so Dr. Dempsey, this is known as the acronym SOT.
SOT.
Okay.
Yep. Now, what I've been doing is combining it in a sort of way, in a I guess a sequential way, I should say, with therapeutic plasma exchange. And therapeutic plasma exchange is basically where you take out plasma. So in your blood, you have red cells, white cells, platelets, and they're hanging out in plasma. And the plasma is where your antibodies are. Your plasma is where your sort of, we call them senescent cells, cells that are, they're not dying, but they're not living. They're not doing good stuff. There's garbage. There's stuff that the cells have released that is the body's not able to clear. And the accumulation of all these things, including the infections that are also in the blood, really can interfere with the ability for patients to recover. So if you can you pull out the blood, you separate it. This is a special machine that does that, of course. You separate it, you get rid of the plasma, you collect the plasma, and that's where all the garbage is. And you infuse back something called albumin, which sort of replaces one-to-one what you've taken out of your body. So you still have enough fluid going through. And the albumin allows the body, it's actually anti-inflammatory, and sometimes we use a little bit of IVIG at the end. The point is that with plasmapheresis, what we're able to do is a lot of these patients are toxic. They're toxic from plastics, they're toxic from mold. They've got a lot of garbage in their system. They've got the bugs that are not being cleared. They have biofilm. There's lots of things that are, I failed to mention the biofilm, but that's a huge part. So when you can take all that out and you do that repeatedly and you sort of set the stage for true treatment and healing, then you go in with the SOT so that it then can target whatever's left. I think you can do a better job at lowering the load, right? So that's what we're doing in my practice, and I'm really excited about it. I mean, I think it's I'm not going to go out here and say it cures everyone. I would never say that. I don't think it's right for everyone. Obviously, it's involved. There are a lot of things to consider, but I'm excited about that, and I promise to publish on it because I really think we need to understand the data and whether this is really going to be the game changer.
Please do publish on that. And I'm going to look, if I had a watch, I'd be looking at my watch so we can give you some time limitations on having this publication available to folks. So I want to ask you about one more thing before we take this conversation to the cellular level with Dr. Stein. And can you talk about how you're using drugs like Ozempic to modulate the immune system and how that gave you a new set of tools and the different types of, is it GLP-1? What are the different types and how you're using one type versus another? That was another fascinating brain melt that you had given me when we were together last.
Thank you. So I did publish with several other with co-authors the utility of using GLP-1 receptor agonists for mast cell activation syndrome. So basically, what happened was we're always looking for things that are going to calm these mast cells down, and these mast cells have these different receptors on their surface that read the environment, and they happen to have receptors for GLP and GIP. These are these various hormones that are produced by the intestines in response to food. I started noticing very early on that patients who were going on these drugs for weight loss were also noticing improvements in their immune system. I was noticing that they were getting improvements in their brain function, cognitive function, memory. Like there were just things that were getting better that I didn't anticipate. And so that made me really wonder about the patients who are not needing to lose weight, but rather we need to reduce inflammation. So we started, and lots of my colleagues have started doing the same thing. Microdosing. What if you use a small dose of these drugs, right? So Ozempic is semaglutide. It's a GLP-1. Tirzepatide so Ozempic and Wegovy are the same drug. One's for diabetes, one's for obesity, but it's the same drug. Tirzepatide is in Mounjaro or Zepbound, and it's a GLP-1 and a GIP hormone agonist. So it does two things. That generation of drugs, that's the next generation, seems to have less GI side effects, even though it might still have GI side effects. And then we have a third one coming out any day, I think, like in January, which is a triple agonist, and it's retatrutide. and it's a GLP-1, a GIP, and a glucagon receptor agonist. That's blowing the results out of the water. I mean, it's incredible what people are seeing with that. So these are drugs that are, again, marketed for diabetes, for blood sugar control, for weight, but they're stabilizing mast cells. And we know that mast cells are very much integral to the metabolic system, the endocrine system. So we know that patients with MCAS have insulin resistance, have a lot of issues. It may be working through that mechanism, and maybe working through how it's just binding to the mast cell and telling the mast cell, hey, chill, don't stop, releasing all the chemicals. But it's been incredible. And so what I used to do is I used to try patients on so many different therapies, and then this would be like the last resort. Okay, maybe we'll try a GLP-1. Now it actually is very early in my treatment plan, just because of the results that we're seeing.
That's really exciting, and thank you for sharing that with us. It was an awesome discovery I made during the last conference. I'm sure folks are really excited to hear about these really creative tools that you're using. So now, one of the things I really love about this panel that ILADS has assembled is that we have a diversity of backgrounds. We have two allopathic doctors, one teaching us the importance of the mental health piece, another doctor teaching us certainly the importance of the immune system. We have our brilliant engineer who gave us the foundation for testing and what we need to do now with testing. And now we have a naturopathic doctor. And I just want to share a little bit about Dr. Stein from my perspective. So one of the things that was really frustrating for me as an early podcaster is that I was losing hope. Now I feel very differently about that 5 years later, because there have been so many powerful improvements that we see in the tools that doctors have to help us through this challenge. But one of the things that caused me to get over that challenge that I had as an early podcaster with a loss of hope is I started to meet these patients who were inspired to go to medical school and use their journey as an inspiration for becoming a doctor to treat other people. And I'm always looking for these young doctors who are taking that step from patient to doctor. And my favorite young doctor last year at ILADS was Dr. Melanie Stein, who's just an awesome servant of people in this community, and who's used her journey as a patient to ultimately become one of the best young doctors in this community. And she's actually written a really powerful book on cellular health. And I thought it would be a perfect way of winding down the panel discussion by having Dr. Stein talk about her book and talk about the discovery she's made about cellular health and how that's an important foundational approach to understanding how to heal from Lyme disease.
Yeah, thanks, Rich. That's a great introduction. And I think that really everyone laid the perfect foundation to come into cellular health, because at the end of the day, every process in the body comes down to our cells. We have trillions of cells in our body, and every day they're making energy and they're trying to communicate messages. And in order to communicate those messages, we need to make hormones, proteins, neurotransmitters, and energy. And what Lyme does is Lyme damages our cells, and it damages those cellular functions so that our immune system can't work and communication can't occur. And what's probably the most important way that it damages it is it damages our cell membranes. So when we think about our cell membranes, we think about this structure around the cell, right? And it's so important at regulating what's able to come in and what's able to come out. And so what Lyme does is it creates a lot of different inflammation in the body, and by doing so, damages the cell membranes so that the nutrients we need for different processes to happen cannot happen. Lyme and other vector-borne illnesses and viruses, parasites can enter cells easier, but then we can't get toxins and waste out as we would like to. No, sorry. And so, when that happens, not only do we break down what can come in and out of the cell, but we're breaking down communication pathways also. So we're not able to send alert signals, and we're not able to send our immune system signals as well. And then inside the cell at this deeper level, we have mitochondria. And mitochondria are so important because they're the powerhouse of the cell, right? All of our energy is created in the mitochondria, and they make these specialized structures called ATP. And ATP has everything to do with energy. Everything that happens in our body depends on ATP. And so when Lyme disease comes in and it wreaks havoc on all of our cells, wreaks havoc on our cell membranes, we're not able to heal. And so that's what I've really found is, one of the biggest missing pieces is we're not treating the terrain. We're killing the infection, and that's great. But what happens when we send all these signals and we've taken care of infection, but our cellular makeup's still not able to heal?
So we're hearing the same message over and over and over again, and that is that we should focus on the bugs, but it's really the terrain, right? It's our emotional terrain and our physical terrain that's going to cause us to be successful. And it's sort of the way we describe it at Tech Boot Camp is that prehabilitation, where we understand that we are suffering from toxicity. We are emotionally toxic and we're physiologically toxic. And as a result of being physically and emotionally toxic, we are now chronically ill. So can you talk with folks about some treatment modalities that you're using in your practice, Dr. Stein, that are helping you to treat folks on a cellular level and what impact that's having on your patient population?
Yeah. Yeah. So, when we think about the cell, we think about that it's made up of different fats, and it's actually those fats that are getting damaged in the cell membrane. And so in order to heal the cell membrane, we need to not only replenish those fats with all of the different healthy phospholipids, phosphatidylserine, ethanolamine, phosphatidylcholine, but we need to get rid of all of the oxidative stress and all of the inflammation happening in the body to begin with. So at our clinic, we use cell membrane therapy, which is a specialized IV therapy specifically based on a patient's lipidomic testing to not only replenish those cell membranes with IV phosphatidylcholine, oral fish oils, oral omega fatty acids, and other, linoleic acids, but different mitochondrial repair supports as well, so that we're able to not only just turn the cell back on and get it to start communicating and working again, but we're telling the body it's safe again. And that's something, that I didn't touch on a minute ago that I think is really important is, long after the infection might be gone, our cells get stuck in what's called the cell danger response. And so it's basically like saying there's this alarm on all the time, and the cells are saying, hey, please help me. I need help. And so instead of taking an input where we say, hey, here's some of the best herbs, here's some of the best antibiotics, please go and kill this infection, the cell says, what do I do? Because I just need help. And so by really healing the cells and with the use, right, of these phospholipids, and we use a lot of butyrate in our practice to help get the toxic fat out of the cell that is a consequence of that inflammation, we're able to tell this body that it's safe again and able to get the cells to turn back on, start making energy again, start making hormones and proteins again.
All right. So what I'd like to do before we open up, and we have a lot of questions in the Q&A section that I'd like to run by you folks, but I want to give each of our panelists an opportunity to just maybe 2 minutes to address any new thoughts that they have based on what the other panelists had said. So Dr. Winfrey, I'm going to come back to you. Any new thoughts that you'd like to share? One of the things that was jumping out at me as I was listening to your fellow panelists is that when you're suffering from chronic Lyme disease, your body is screaming, your brain is screaming, everything is screaming. And Dr. Stein was talking about the communication between the mitochondria and the immune system. She was talking about the cell danger response. And what we didn't really get to talk about is the amygdala and how the smoke detector in our brain is also going to be going off. So we just have all these alarms going off when we're suffering from chronic Lyme. So can you talk a little bit about the brain element of this? Because we have two sides of it's all about the cells or it's all about the brain. So it's really exciting to have this combined conversation.
Well, to some degree, she's right. And these different components of the cell come together, the organelles within them, and then they make up a cell, and those cells then eventually make up an organ, and then those organs then make up systems, and then those systems create connections, with the brain, and they communicate. And so all that information is flowing to the brain and back down, and the limbic system is sensing all that sort of information. And so earlier we were talking about the parasympathetics, and so I think that sometimes in our treatment process, what we've talked a lot about here today is how you set up treatment, and sometimes there's a rush to go in and start killing. And before we get there, you call it prehabilitation. We want to really set the terrain, and so one of the first areas we do want to assess is the limbic system, what is assessing threat. And we want to make sure that that's stabilized and calmed down. And then we go into the autonomic nervous system, making sure that that's regulated. And then making sure that we are able to make ATP and have the mitochondria upregulated and things of that nature. And so we have to, in some degree, restore some order and some stability, and regulation becomes before eradication. And so the nervous system needs predictability and gentleness and trust to be able to heal. And so we want to really calm down those critical systems so we can eventually get to growth and resilience. And I think to some degree, where we was talking earlier about the mental piece, one of the things I was reflecting upon this week was 1 Corinthians 13, the chapter in the Bible that talks a lot about love and how love is patient and love is kind, and it does not envy, it does not boast, it's not proud, does not dishonor, it's not self-seeking, it's not easily angered, it doesn't keep record of wrongs. That is a very sympathetic way of living. And to some degree, where people with Lyme disease and these chronic infections are really struggling with, what we really here on earth is to really have loving relationships. That's what all of this matters. So all this biochemical analysis that we're talking about should get us to a place where we have loving relationships. That's what matters most. Because when we die, that's what we're going to be focusing on most. That's what we're going to want us around. We're not going to be thinking about all the degrees on the wall. We're not going to be thinking about the money. We're not going to be thinking about our jobs. We're going to be thinking about those loving relationships. And when we are in this fight or flight survival mode because of these illnesses and the impact it's having on us, we really can't adequately get into a state to love. And so for those that are married, those in a relationship, and they're saying, I love you, but your body's chronically stressed, it's not the deepest form of love that you can get to. And so the work that we're trying to do is ultimately getting our body to that sort of place so that we can deeply love and finally be known and understood and have that rich intimacy.
I love that. I love that. And of course, one of the challenges that we've certainly observed at Tick Boot Camp is how to define a relationship, right? And what we were created to be is to be interdependent. We're not supposed to be independent, where we're by ourselves. We're not supposed to be dependent, which is one of the most painful parts of a Lyme disease journey. We feel like we're dependent. We're still supposed to be interdependent, where we're serving other people and other people are serving us. And that is certainly the very definition of love. So I love that you took it that way. So, Nicole Bell, any thoughts occur to you after listening to all your brilliant co-panelists?
Yeah, I mean, I think I want to echo, Dr. Dempsey's the patience that everybody's on fire, right? And actually, somebody gave words to it at the Lyme Bites conference this year. It was like, universal responders, right? They're just lighting up everything. And I was like, yes. And we see this because people we have patients that reach out to us, we have practitioners that reach out to us, and we see these labs and we're like, where our co-infections are common, I don't think you have these 10 tick-borne infections, because looking at the ecology of it, right, you would have to travel to all these different regions and different things. And so it's like there's something else going on. And so that's a very important complexity that I think it's important for patients and providers to understand is that, just because you have the test result, you have to understand exactly what it means and use that accordingly. And I think all the discussion on the ways to get better is and addressing the emotional components and the cellular components. We work really closely with providers, and the ones that are having the most success, they're definitely integrating all that into their practice. So I can't comment on any of the treatment modalities because that's way beyond my pay grade, but it's been fantastic to hear from everyone.
Dr. Dempsey, any thoughts before we go to the Q&A?
Yeah, I want to sort of echo what Dr. Winfrey has been talking about and say that, I talked a lot about medications and other treatments for the immune system and mast cell activation syndrome, but probably the most important treatment, and that I really start everyone on right from the first visit, is limbic retraining, or something to reset and balance the autonomic nervous system, the sympathetic and the parasympathetic that Dr. Winfrey so beautifully and elegantly explained. It is really the first step in healing, because a dysfunctional nervous system, being in fight or flight all the time, is going to affect the immune system, is going to affect the ability for the body to function, to heal, for the cells to function. I love what Dr. Stein talked about. And, in order for everything to work, right, we need to be in a different state of mind and state of body. So, Dolan?
One of the things that I discovered again in my own personal journey and listening to many of the folks we interviewed is that there's a physiological and an emotional component to being triggered into fight or flight. So Dr. Dempsey, can you talk to us a little bit about the HPA axis and the physiological elements of the HPA axis and the stress hormones that are triggered and the need to modulate your cortisol receptors? And then, of course, the emotional piece to that, where you are now triggered into fight or flight. So give us some insight into the HPA axis and the importance of modulating the HPA axis both physiologically and emotionally.
Yeah, so right. The HPA axis is the hypothalamus, the pituitary, and the adrenal glands, HPA. And there's, in our body, we have all these feedback loops, where different organs are talking to other organs. And in the endocrine system, the hypothalamus will produce certain hormones, tell the pituitary to produce certain hormones, will tell the adrenal glands to produce certain hormones. And the end result in the adrenal gland are these hormones like cortisol, adrenaline, epinephrine, norepinephrine. There are a lot of things that the adrenal glands produce, DHEA, and others. So the problem is that it's in a perfect world where we are all sleeping exactly 9 hours a night, and we have no stress, and we have everything, right? Everything is working perfectly. And let's just say in prehistoric times when there was less stress, I don't know, the body would produce cortisol in the right time of day. The body would respond, everything would be, appropriate. But we're living in, I kind of think about it. I use this analogy with patients, basically back in the day, it would be like a bear would come or a lion would come, and we'd have like fight or flight, but then you'd have like weeks when nothing came and you were fine. And here it's like that lion or that tiger or bear is chasing you literally all day long. And it's basically causing this dysfunction in that axis, and basically in lots of other organs as well. And so now the adrenal glands are either producing too much or too little, or are producing it at times that are inappropriate. So, you're supposed to have cortisol highest in the morning. That's what sort of wakes you up, gets you out of bed. Through the day, you have these little spikes, and they get lower and lower and lower. And, just you still produce it, always have a little bit in your blood, but but it's enough so that you can go to sleep at night. And then in the morning, again, it goes up. Our chronically ill patients, they can't get up in the morning. They're exhausted, right? Their cortisol levels are low in the morning. But they often say, I mean, how many people out there say, I get wired. I get at 10 p.m. I can accomplish more than I can all day long.
Tired but wired.
Tired but wired. And because their cortisol now is higher at night instead of in the morning. And so now there's this flip-flop. And that's really a hard state, because now the cortisol is higher at a time when the body needs to recover.
I think it's a really, really great way of explaining that and helping folks understand what is going on, not just emotionally, but physiologically, and how we have to strike that balance and make sure we both have our mental health professionals and our allopathic or naturopathic professionals who are helping us with our physiological piece. So, Dr. Stein, I'm going to give you an opportunity. You are our last speaker, but in the event that something was triggered in the last couple of conversations, I want to give you an opportunity before we turn to questions. And I might ask our panelists if you could just stay with us for another 12 minutes, and we'll try to get to some of the questions that are in the Q&A.
Yeah, I mean, I think I just want to speak to really how we just designed this tonight, right? And that it's really about treating the terrain. And so, I know just very well firsthand that sometimes treating Lyme, it's like, okay, well, now I need to treat my hormones. Now I need to treat my limbic system. Now I'm trying to regulate my sleep and my thyroid and what's happening with all of this autoimmunity. And it really is this multi-system complex chronic illness. And, at the end of the day, there is not a one size fits all treatment for this. And I think that's why we're all here today talking about these different mechanisms in which Lyme and other vector-borne illnesses attack our immune system and our cells and our brain. And so I just really want to give hope to everyone that, people, you can get better and your body wants to heal. And, there's just some incredible doctors out here that are doing some amazing work. And so, stick with it. It can feel hard at times, but, with the right support and with, the right puzzle pieces being put together, the body is resilient and long-term health is possible.
Thank you. Sorry. So we have 11 minutes left before we wind down. And what I'm going to do is go to the Q&A, and I'll start to read some of the questions and ask any of you who feel called to answer the question, please do. So Kathy Swift asks, do you know of any connection between Lyme and blood cancers? Anyone wants to field that question?
So I do know in the research that there's a link between Bartonella and like canine hemangiosarcomas. There's also been research that's linking it to inflammatory breast cancer. And at this point, they're still trying to do more research to figure out, is it causal? Is it kind of just happening along with, right? But a lot of that effort is still ongoing. And I know that Eva Shappy has also done some research work on the link between Lyme disease and breast cancer and so forth. But it's really an emerging area that needs further study, right? And I think that that's the case for a lot of named condition. Alzheimer's is one that's near and dear to my heart because of my husband's condition, but you could go through a whole list of, chronic fatigue syndrome, fibromyalgia, ME/CFS, like there's all sorts of named conditions that are just really amalgamations of symptoms. But cancer is one that there's definitely a lot of smoke, right, that needs to be studied in more detail.
So Kathy, I would also recommend that you listen to the interview that we at the bootcamp did with Dr. Eva Shafi, and she spoke in great detail about the work she's doing connecting Lyme disease and breast cancer, and she's also spent a fair amount of time reviewing Dr. McDonald's work on the connection between Lyme disease and some forms of brain cancer, specifically multiform glioblastoma. So I hope you would tune into that episode for more. And thank you, Nicole, for the great answer. So the next question I'm going to read is, Eric, I apologize for butchering your name. Eric Gervaris asks, is Lyme and co-infection known to cause herniated disc in the literature? I had a herniated disc at L3, L4, 2 years post-infection. A mortic change type 1 was observed on the vertebra also. Does anyone have any insight into helping answer that question? Anyone come across anything in the literature?
The only thing I'll say, not in the literature, not specifically with herniated discs, but I will say that in patients who have mast cell activation syndrome, they often have other associated comorbidities, and one of them is changes in connective tissue. We also see this directly from the organisms, the pathogens themselves. We know that Lyme, Bartonella can actually also directly damage collagen
Yes.
in the connective tissue, as can the mast cells that are in the same places as those pathogens. So I think there's a a relationship there. And so when I think about the vertebrae and the spine, I think about patients who have instability sometimes in their spine because of the changes in the connective tissue and the collagen. So I don't know if that's something that could lead to a herniated disc. I would assume it could, or something like that. Again, that's just conjecture, but I just did think it was important to mention the connective tissue issue.
Yeah, so in following up with that, Dr. Dempsey, one of the things that we had observed during our interviews is many of the people who are chronically ill had EDS or were diagnosed with EDS.
Yeah,
Now, do you believe that Lyme causes EDS, or do you think EDS makes you more vulnerable to chronic illness from Lyme? What are your thoughts on that?
or both.
Okay, or
I
both.
think okay. No, I think that I know I have a different lens, a slightly different lens than others. But what I think is happening is that the infections themselves have mechanisms to break down connective tissue, making them more hypermobile, or again, causing this EDS-like picture. But the mast cells that are fighting those infections are releasing also lots of different enzymes like elastase that's breaking down the elastin, that's also making the connective tissue stretchier and not as stable. So I think it's a combination of factors causing that. But I also think that having this immune dysfunction, mast cell activation syndrome with connective tissue issues probably does affect the ability to fight these infections off. I say very often, right? I think a lot of people out there have Lyme disease, have Bartonella, have Babesia, but not everyone is sick. So there must be something about why people are sick. And maybe one component is that immune dysfunction, and maybe the connective tissue issue, EDS, is also a risk factor.
But I think that most of us can manage the microbes until we become toxic, right? And then that's what ultimately causes the boiling over of the pot, to use Dr. Rawls's metaphor, and ultimately that's where we find ourselves, right? So, Dr. Stein, you have any thoughts to follow up on Dr. Dempsey's observation?
I mean, I think the same thing, right? Is it's probably both. We know very well, especially with Bartonella, the way it works on cartilage and connective tissue also. And so, I think it goes both ways. I think that these infections create inflammation, which further damages the ligaments, the connective tissue, the cartilage, and then also, whether or not, as we are more vulnerable, right? These infections like to go to vulnerable areas. So if there's already hypermobility and ligament laxicity, are these infections going to go in and then attack those areas as well?
Perfect. Okay. Ali Chang asks, hi, can you explain why a patient may test positive for brucella, presumed false positive, and how that could be a Lyme indication? Dr. Dempsey?
Yeah.
Well, I guess, well, there are some in the community that are suggesting that a positive result in one infection could be a marker of another infection. I don't know that I've heard that specifically with brucella, but I've heard it, let's say they have an antibody, an IgM antibody to Lyme. Maybe that really represents Bartonella. I've heard these various theories, but I don't know if I could, say directly that it indicates Lyme. I don't know what the other.
Nicole?
And yeah, and I would add that again, the test matters, right? So if you're testing for antibodies, antibodies have a lot of cross-reactivity, right? And so how specific is that antibody and what they're testing for in that particular lab for that particular pathogen? And so there's a lot of cross-reactivity. Brucella is relatively close, to Bartonella, and there's things that can cross-react. I mean, example I've talked about, even within, it can be across species or within species. I've talked about my husband's case that he tested positive with serology for Babesia duncani, and it didn't seem to fit. And sure enough, he actually had Babesia oticolii, which is a different type of species and behaves differently in the body. And I think depending on the lab that you're using, depending on how they do their tests, it could be a cross-reactivity issue. But I think I haven't heard of anything specific. I mean, a lot of times, again, these infections, like you said, Rich, right? A lot of people get exposed to Bartonella, right? Like cats have it, our household cats have it, and a lot of people have it and they're completely healthy. But then the pot boils over, you reach the tipping point, whatever analogy that you want to use, you get another infection, and then all of a sudden, things start to flare. And so I don't think they're directly related, but I do think that they're kind of a corollary. And then you do have to be careful about the test and what it's actually testing and reporting.
Dr. Winfrey, any thoughts from you?
On that particular topic?
Yes.
Oh, I agree with them. I support them.
Okay. So I'm going to ask one final question before I allow our brilliant panelists to go back to their families. Roberta McKnight asks, is anyone testing nasal drainage? This may be most fruitful. Any thoughts on nasal drainage from any of our panelists? No thoughts on nasal drainage. Okay, well then,
Well, I'll
okay,
say, I mean, I don't test nasal drainage per se, but I do nasal swabs
okay.
in patients who have had, let's say, mold exposure. We sometimes will see colonization of certain staph species, sometimes mold in the nasal cavities. Sometimes that affects the ability to fight the other infections, and sometimes that has to be addressed. So that is, yeah, that is one area, but not for Lyme. I guess they're asking about nasal sampling for Lyme disease.
Or
Nicole,
maybe,
you want,
I
you
haven't
want
I
to?
haven't seen a lot of evidence in the literature for kind of, for the three Bs, for example, about residents kind of within the nasal passages and so forth. So there's some work that I've seen and, looking at saliva, for certain pathogens and so forth. But I think, again, we kind of rely upon urine for the Lyme tests, and then I think, blood for the three Bs, including the relapsing fever Borrelia, so that that it really depends on the microbiology of the pathogen and is it present and how are you detecting it?
you think it's possible she may be asking a question about whether or not using different type of nasal drainage tools might be helpful in healing? Like I don't know, like an as a EpiPod is it called? I don't know what I've never used it,
Or
but
Oh, like a
neti pot?
neti pod or
neti pot. Thank you,
yeah.
neti pot. You think that might do any of you know anything about using a neti pot as a treatment tool?
I mean, I think to piggyback off what like Dr. Dempsey was saying, I think that nasal colonization can have a big impact on the ability to eradicate infections and also on our brain health as well. It's a direct pathway. And so I do a lot of different sinus irrigation formulas, nebulized formulas to make sure that we are treating something that might be colonized in the sinuses to help with that whole, whole body train approach. I'm not sure if that's the question, but
Okay, well, thank you all. So we are now 90 minutes in, and I did promise the folks at ILADS that I would not take too much of your time away from your families. So I want to thank each of you for spending the last hour and a half with this community and all the people who will be watching this when we post it on video and audio formats. I want to thank ILADS for their approach to now creating community service content. This is new, so I have to compliment Ali Moresco and Sarah Quinlan for taking this approach to not just training the best and the brightest doctors in the Lyme disease community, but also giving folks in the community generally access to these doctors that they may not otherwise have access to. So thank you all for attending this event, and I hope you found it all fruitful. Thank you all, and good night.